are not familiar with the Illinois Science Council, we are an independent, i.e. we're not funded by the state of Illinois. We are an independent nonprofit organization with a mission of promoting awareness and appreciation of science and technology to the adult public. As a matter of fact, we do public programs like Author Talks and hands-on programs like Chemistry of Beer and Chemistry of Chocolate and programs on nature and physics and all sorts of fun stuff like that. So if you're not already on our mailing list, why not? You should be. You can sign up for that out in the lobby afterwards, and also you should be liking us on Facebook and following us on Twitter and all those fun things. So, I hate that I have to say this. Please turn off your communications devices. This program is being recorded tonight for WBEZ as part of Chicago Amplified. Also, Dr. Shubin will be taking questions, and his books are for sale, and he will be signing copies of those in the lobby afterwards, courtesy of Seminary Co-op Bookstore. So, Dr. Neil Shubin, educated at Columbia, Harvard, and the University of California at Berkeley. Dr. Shubin is the Robert Bensley Distinguished Service Professor of Anatomy and Associate Dean of Biological Sciences at the University of Chicago. He was elected to the National Academy of Sciences in 2011 and served for 3 years as Provost of the Field Museum. His research is on the evolutionary origin of anatomical features of animals. He's conducted his work in Greenland, China, Canada, much of North America, and Africa. His most recent discovery, Tiktaalik rosé, has been dubbed the missing link between fish and land animals, also known as the fishapod, which is my favorite new word. It is the only fossil to appear twice on the Stephen Colbert Show. So, with that, please give a warm welcome to Dr. Neil Shubin.
I've given a fair number of talks in the last month and a half, but this one's special because it's home, it's in Chicago. Thank you for coming out. And yeah, we're going to talk about that Colbert fossil in a few minutes. I'll show you a little bit about that. It's the one thing it's known for, not because it's important, but because it's been on the Colbert Report for 2 times. As you heard, I'm a paleontologist. And as a paleontologist, I dig in the rocks. I look inside the rocks to understand the past. And I do my work with the knowledge and the conviction that understanding history, understanding the past, changes the way you see the world. It changes the way you see the present. Because when you see history, when you see the history of bodies, of rocks, of planets, of stars, you begin to see connections that would be invisible to you otherwise. And where do those connections begin? They begin in history. They begin in the shared history that we people share with everything else in the universe. And it's a beautiful story. Because once you see DNA, once you see fossils, once you learn planetary formation and the cosmos and how it came about, what you see is that in every gene, cell, and organ of our bodies, we have over 3 billion years of the history of life. A shared history with the planet that extends over 4.5 billion years. A shared history with the solar system. And inside us are even artifacts of over 13 billion years of the history of the universe itself. Layer after layer of history exists inside of us. And for me as a scientist, it's a great privilege to be able to discover that history. We don't just happen on it randomly. We find it. We actually look for it. We plan expeditions or look at the stars in ways that give us clues to this vast past, much of which is contained inside in our own bodies. So what we're going to do tonight is we're going to see these layers of history inside of us. We're going to see fossils that connect us to the rest of life on the planet. Then we're going to start at the Big Bang, and we're going to begin about 13.7 billion years ago and go to the present, and to show how artifacts of the formation of the universe, and indeed the solar system itself, are inside our very makeup. It's a tall order. We only have 45 minutes or so let's get going. We're going to begin with our connections to the rest of life on our planet. Because as a paleontologist, I think about this all the time. The key thing here is learning to be a paleontologist, learning to be a cosmologist, means learning to see in new ways, learning to see the past. And how we do that is we make discoveries. We use previous discoveries to leverage future ones. So what I want to do is talk a little about how we go about finding fossils. Because fossils are my primary window to looking in the past. And then we're going to generalize from there. My intellectual beginnings, if you will, really had their start in my second year of graduate school. Like every graduate student, I had no idea what I wanted to do. I knew I was interested in the field of paleontology, but I was really nervous about finding a dissertation that I could peg my hat on. And in my second year of graduate school, the professor showed a slide which really changed me and changed my life, and it became the problem that I've been working on, the scientific puzzle that has occupied my brain for the last 30 years or so. This is the diagram he showed. I know I seem like a crazy person that this would change my life, but it really is a life-changing diagram for me. Because what it is what we knew about the transition from life in water to life on land, what we knew about this in the mid-late 80s. And what you see is a textbook diagram that he showed, and it shows a fish on top. This is a cartoon of a fossil fish, a creature that existed about 390 million years ago. And it's a lobe-finned fish. On the bottom is a cartoon of what we knew about at the time of an early land-living creature, so-called tetrapod, with fingers and toes. And I remember looking at this diagram and thinking to myself, golly, what a first-class scientific puzzle. Think about how big this change is. Life in water to life on land, virtually everything has to change. There's an entirely different ecosystem, entirely different physiology. And these creatures look very different. In fact, I could spend the entire talk talking about the differences between fish and land-living creatures. You can see their heads are different. Fish have fins, land-living creatures have limbs. And my quest really became to find, say, a flat-headed fish with fins, some sort of cross, an intermediate between these two creatures. So what did I do? I pulled out, just like every paleontologist had done for a century or more, is I pulled out the paleontological playbook to find new fossils. And what do when we find new fossils? Is we look for places in the world that have rocks with three properties. The first is we look for places in the world that have rocks of the right age to answer whatever question we're interested in. Remember I told you that these guys on top are about 390, these guys, the limbs on the bottom, are about 365. So we want to have places in the world that are in that window of time, preferably about 375 million years old. The other thing is you need rocks of the right age, but you also need rocks of the right type to hold fossils. Not every kind of rock preserves fossils, either because the rocks have been baked or squeezed or pressed, they're metamorphic rocks, or because the creatures didn't live in the environment that the rock was formed in. So as a paleontologist, you begin to learn likely and unlikely kinds of rocks to hold fossils. And so, but there's a third criterion that's really important. It does me no good if my wonderful rocks of the right age and the right type are buried 5 miles underground. Those rocks have to be exposed to the surface. And the reason for that is, not because we're lazy, but it's because when bones weather out on the surface, when we have lots of bedrock to look at on the surface, we increase our odds of finding fossils. And just digging blindly in the dirt would do us no good. So we look for places in the world that have rocks of the right age, rocks of the right type, and rocks that are exposed to the surface. For this particular thing, it really came down to finding rocks about 365, 375 million years old. Rocks that were formed in ancient delta systems. We knew that from previous work. And rocks that are in deserts or exposed to the surface. There's another criterion, which is convenience and lack of money, in my case. I took my first academic job in Philadelphia at the University of Pennsylvania, and I didn't have a lot of money, or as a junior scientist, I couldn't afford a lot of risk to find new fossils. So I dug out a geological map of the state of Pennsylvania, and it turns out that Pennsylvania is just loaded with Devonian age rocks, which are rocks more or less of the right age. Not only that, it's loaded with Devonian age rocks that were formed in ancient delta systems. But Pennsylvania, as you probably know, is not a desert. The bedrock's not exposed at the surface. So what my colleague, Ted Deschler, who's still working in Philadelphia, what Ted and I did, was we ended up following the Pennsylvania Department of Transportation around as they made new roads. Because what happened is, PennDOT underwent a huge building program in the late 80s and early 90s. And I saw that slide, which I showed you the cartoon in the late 80s. But by the 90s, when I moved to Philadelphia, they were opening up road cuts in central Pennsylvania right through primo Devonian rock. So, as soon as they cut through the rock, Ted and I were there, and we were looking for bones. And what you see here is, it's called Red Hill, Pennsylvania. We're not clever namers. It's Red Rock on Red Hill. It's about an hour north of State College, Pennsylvania. Here's a human being for scale, right there. And here you see the strata, or the layers, that these Devonian age rocks formed in. And each of those layers reflects the life of an ancient stream. If you look at them in cross-section, they're sort of lenticular. And they show like a bed, like a stream has, with cobbles in the center and fine grains on the outside. Anyway, those are the geological conditions. But this was paleontological heaven for us in the mid-90s. Right where that guy is standing was a pocket of rock that contained numerous bones of all kinds of different types of fish. Indeed, what you see here is we started early on, started to pull out limb bones of some early land-living amphibian creatures. We pulled out portions of the skulls, portions of the shoulders, portions of their leg bones. It was a wonderful opportunity to find early land-living creatures. And working with National Geographic in the mid-late 90s, we were able to reconstruct what that road cut looked like in these ancient times. What you see is it was an ancient stream, freshwater stream, with some of the earliest forests. We were finding pieces of trees. There were fish, some of them 16 feet long, with teeth the size of railroad spikes. I mean, giant monsters were coming out of this road cut in Pennsylvania. I mean, trucks would be whizzing by, pulling the monsters out. Armored fish, and many types of limbed fish. This is what I mean about how history gives you a knowledge of history, changes the way you see the world. If you drive by that road cut, what would you see? Just a cliff of rock. But inside that rock is this ancient world from rocks, from an age of 365 million years ago. Now, this talks about your inner universe, and we're not getting there. Well, how we get there is to try to find that intermediate first and our connection to the rest of life on our planet. Turns out that these rocks, while we were finding lots of fossils, were actually no good for finding that intermediate, like that flat-headed fish with fins I was telling you about earlier. Turns out we were in rocks that were about 10 million years too young to answer the question. And we knew that because other colleagues working around the world had found bits and pieces of tantalizing creatures in rocks about 375 million years old. And they found some of these creatures in Quebec and in Eastern Europe. So Ted and I decided, well, we've got to start again. So we pulled out the paleontological playbook again and had a plan to work in Brazil. We had a plan to work in Alaska. Everything changed one day in my office in Philadelphia in 1998 when Ted and I had an argument. And to settle the argument, I pulled out my college geology textbook and settled the argument with some minutiae of geology. And after the debate was over, or the argument was over, I'm thumbing through, we're just chewing the fat, thumbing through. And I see yet another diagram that changes my life. This is a diagram that launched almost 13 years of expeditions. This is it. A diagram from a college geology textbook, which enabled us to make arguably the most significant paleontological discovery of my entire career. I'm going to spend a little time on it because it shows you exactly what we look for when we go to find fossils. So here it says Upper Devonian sedimentary facies, which means rocks of the right age window, to answer the question, sedimentary rocks, which could be of the right type. And what you see here is a map of North America, right? There's the United States in the middle, there's Canada up north, and superimposed on that map is an interpretation of the setting in which the rocks, the Devonian age rocks, formed. So the key to this is that this kind of hashwork here reflects that the western part of North America contains Devonian age rocks that were formed in an ancient ocean, in marine systems. But these authors identified 3 areas, shown in red, where the Devonian age rocks were formed in ancient delta systems, like the Amazon Delta today. The first one I knew about. This is that Catskill, those Catskill rocks. Remember the rocks I've been showing you for the last couple minutes? Those were the rocks we were working on. So this sort of confirmed that they sort of knew at least part of what they were talking about. There's a second set of rocks here in East Greenland. These we knew about too because these were really well studied. These rocks produced, remember that cartoon I showed you of that flat-headed creature with limbs? That cartoon that motivated this whole thing? That was discovered by Danes and Swedes working in the 20s and 30s in East Greenland. I think you see where I'm going. Extending 1,500 kilometers east to west across the Canadian Arctic, where Devonian age rocks formed in Amazon Delta kind of systems of the right environment, of the right type, to produce fossils. Furthermore, they were the right age. These were 375 million years old, right at the exact time period where you hope to be able to find, say, a flat-headed fish with fins. So Ted and I, this all happened one morning in my office in Philadelphia. We were so excited. We went out for lunch for Chinese food to celebrate. I had my Kung Pao chicken or whatever. But I had a fortune cookie. And I opened the fortune cookie and I read it and it said, soon you will be at the top of the world. Okay, we're at the 10 years. So it took us about a year and a half from that fortune cookie to get to the Canadian Arctic. And the reason why it's tough is because we have to move around with fixed-wing aircraft and helicopters. We're beyond the tank of gas of a helicopter, so these fixed-wing rotary aircraft land on the tundra to bring the gas. Anyway, it took us a number of years. The Canadian Arctic is a big place. Fossils are very small. It took us a number of years to winnow it down to a particular spot. One of our college students who joined us on the expeditions found a layer which was covered with fish bones. And then we exposed that layer. Here's Ted right here with a jackhammer. We exposed that layer, and it turns out all those fish bones at the surface were produced by skeleton after skeleton of fossil fish piled one on top of the other. So it's really remarkable. We worked this for about a year, for one summer, not finding anything new. And, but then one day, everything changed, but this time not with the diagram. This time, when this colleague here, a guy named Steve Gatesy, was removing rock from this layer. We were all working next to each other on the layer. And he pulls a rock from right here. And he says, "Hey, Neil, what's this?" I ran over there, and as soon as I saw this, I knew we had found exactly what we had been looking for. We had spent the last 6 years, countless dollars, numerous sprained ligaments. What we have here, see, here's the red stuff. This is Devonian rock. But then look at this. See, here's a V. See this little V right there? There's a little crack in the V. Turns out that's the lower end of a front end of a jaw. There were a bit of teeth here, so the animal's kind of smiling at you. And here is a snout, upside down. And not a snout of just anything, but it's a snout of a fish. I knew that because of the armor on it. And not just any fish, it's the snout of a flat-headed fish. Remember, you saw the fish with the conical head, the flat head? Here I had a flat-headed fish staring right at me. So we spent a considerable amount of time removing this fish. Comes back at the bottom of a helicopter. There's a first-year graduate student for scale. These things come back to the laboratory where the fossil preparators remove the rock grain by grain. This is Steve's specimen right here. And the rock is removed grain by grain. This took 3 or 4 months. But look what's emerging right here. So here it looks like you have a head. There's one orbiter eye hole. There's another orbiter eye hole. Fast forward another 5 months in 2005. And here you have, look, here's the head emerging. So there's a flat head with eyes on top. Look, there's one piece of shoulder. There's another piece of shoulder. Hey, look here. It looks like there's a neck. Remember I was telling you that learning to see the past means learning to see history. Learning to understand, learning how to read the rocks in this case. And we were looking for some intermediate between a fish that lived in water and a creature with limbs that lived partially on land. Essentially looked for a flat-headed fish with fins. This is Steve's specimen fully prepared out. What you have is like a land-living animal. It has a flat head with eyes on top. It has a neck, like early land-living creatures. But like a fish, it has scales in its back and fins with fin webbing. But when we open up the fins, what do you see? You see bones that correspond to our upper arm, forearm, even parts of our wrist. This is a true mix between land-living creature and fish. I brought the cast you can see on display at the Field Museum, right down the street. This is a cast. This is Steve's specimen here. The first thing we saw, here's that beak right there. This is what was staring at it. So when I knew that, I knew we found what we had been looking for. So you learn to have this cast. You can't break it. It's been in my kid's school many times, so you will not be able to test it by 7-year-olds. As discoverers of the new creature, we were allowed to name it, and so we had a naming project. And we engaged the Inuit Council of Elders, which are these guys right here, to come up with a name that has 2 properties. One that's a name that's meaningful to them and to us, another a name that we could pronounce. And the name of the committee did not lend me a whole lot of confidence that we have a name that we could pronounce. Anyway, it's really frustrating. These guys didn't have any, they're talking about learning to see. These guys didn't have any, they could see snow, they could see ice, but they had no concept for fossils. So I would talk to this guy, this was in 2005, over the phone from Chicago, and him and Reese Fjord, and, the northernmost settlement in the entire planet. And we'd be on the phone, and I was trying to describe this thing. They had no concept for it. I said, well, we found this fish. We found it in the rocks. He said, well, hunters don't find fish in rocks. Well, okay. And I realized they had no concept for that kind of deep past. Eventually, I got very frustrated with me. He said, look, just tell me what it is and where it lives. I said, well, it's a large freshwater fish. He said, why don't you say so? You got yourself a Tiktaalik. I said, a Tiktaalik, what's that? He says, a large freshwater fish. Anyway, I could spend this whole evening talking about this wonderful fish that with lungs and gills, with fins and limbs, with a real mix of these great features that we see in fish and land-living animals, and what it tells us about the transition, and reconstruct it. But the key part for tonight's story is that this is a window on an incredibly vast past that's a part of our own history. Because this event, this transition from life in water to life on land, is not some crazy, esoteric dead end in the history of life on Earth. It's a transition that's embedded in our own bodies. Because this wrist that appears for the first time in Tiktaalik and its evolutionary cousins is something that's to become our wrist. This neck that appears in Tiktaalik and its evolutionary cousins 375 million years ago is something that's to become our own neck. So every time you bend your wrist, every time you bend your neck, you can thank this great moment in the history of our planet when fish evolved to walk on land. Layer after layer of history is embedded in us, and this is just one of those significant layers. How do we know this? We know this from multiple lines of evidence. We know this from the fossils of the fins and skulls that connect fish like Tiktaalik to earlier fish, deeper fish in the tree of life, and humans as well. We can trace these using the fossil record. We could trace these using the DNA record in embryology. When you do this, you see that every gene, cell, and organ of our bodies contains links to other creatures on our planet. And these links are vital to us. We take them for granted in biomedical research. When you think about how important they are, what is the research that is likely to generate cures for human diseases? What is the research that has won Nobel Prizes in medicine or physiology? You know, sea urchins, mice, flies. 2 Nobel Prizes in the last 9 years have gone to 5 scientists working on a little tiny worm the size of a common piece of paper. Yet that little worm is telling us how our genes are turned off in health and disease and how they die in health and disease. It's not too far-fetched to say that many of the cures, which are going to extend and enrich our lives, are going to originally be based on work on flies, worms, and in some cases even fish. That's a powerful statement about how deeply connected and the importance of those deep connections we are to the rest of life on our planet. But that is just the tip of the iceberg. Because when we see who we are as people, when we see the deep history inside our own body, we find artifacts of events that go all the way back to the Big Bang, 13.6, 13.7 billion years ago. Artifacts inside of us are derived from the Big Bang itself, to the formation of the Earth and the formation of the Earth-Moon system, to the origin of our atmosphere, that which we breathe in the Earth, and indeed even to changes in the working of our planet itself. A timeline of artifacts of the history of the universe, the planet, and the solar system are indeed inside of us. And that's what we're going to work on today. And one of the things you begin to see is, as you learn to see the past, what you learn to see is our world, our planet, and our universe is an incredibly dynamic place. That is, if you look in the sky, you see stars, and they flicker at you, right? But that reflects an incredibly dynamic world that lies above your head. That is, looking at the sky, when you learn to see it, it's like seeing a forest. When you go to a forest, you see old trees, you see young trees, you see saplings, you see plants at different stages of their life history. The same is true for the sky. When you learn how to see the sky, you see stars and galaxies at different parts of their lives as physical entities. Even stars that have exploded, remnants of them, as supernovae. That same dynamism is seen in our planet. If you ever get to the top of Mount Everest and you start cracking rocks and you bring your geological hammer, what will you find? You will find marine rocks from an ancient ocean. There is no better statement about the dynamism of our planet than to say at this top of the Earth, right above the famous Hillary Step where climbers traverse each May, lie marine age rocks that were once at the bottom of an ocean. All this dynamism begins by people applying new tools to learn to see the familiar in new ways. Much of our understanding of the Big Bang, at least the initial understandings of the Big Bang and our connections to the universe, really began with folks who began to understand that these flickering lights that lie above our head each night really reflect a vast and ancient universe. This wasn't always known to folks. And one of the biggest events that happened that gave us this window, much like paleontologists who learned to date rocks, came from people who learned to measure the distance of space. And this breakthrough happened, and there's one critical moment. It happened by virtue of what's called the Harvard computers working in around 1910 to 1915. Edward Charles Pickering, who was then the head of the Harvard Observatory, the Harvard Smithsonian Observatory, had a real problem. His observatory was collecting like the raw data of the heavens. They were taking pictures of space, taking pictures of stars and so forth. But with all these pictures, they now had no ability to analyze them. They were collecting more data than they could crunch and analyze. So what he did is he hired a team of people to begin to crunch those data and to begin to make sense of them. And he was famously cheap, and he complained to somebody that, you know, these guys are working for me. You know, I could hire my maid at half the cost and get more work done. Well, it turns out he fell in love with that. He fired all the men, and he hired his maid, Wilhelmina Fleming, to do the work. And she set up the team here, which collectively became known as the Harvard Computers. These people, these women, working with the raw data of astronomy, were some of the greatest astronomers of their age, and indeed of pretty much any age. Indeed, one of them, Henrietta Leavitt, right here, had the breakthrough. Sitting with the raw data that was coming to the Harvard Smithsonian Observatory, she noticed her job was to look at a particular kind of variable star. These are stars that go from bright to dim at a certain known period. She had a breakthrough, a theoretical, where she was able to intuit and calculate a rule that basically showed that the period, the rate, the time period in which this particular kind of variable star went from bright to dim gave you a measure of the star's real brightness. Okay, that sounds like a very esoteric calculation, and indeed in some ways it is. But here's the trick. By knowing the star's real brightness, she was able then to measure that star's distance from the Earth, because the speed of light is a constant. So what did Henrietta Leavitt do? Just by figuring out those stars and calculating this relationship, she gave us a way to measure distance in deep space. This was huge, because now we had a ruler to apply to the sky. Well, what did this mean? Well, it was in the hands of a Chicago guy. This is Edwin Hubble. He was originally a Chicago guy. Moved to California to lead one of the largest telescopes of his day, in fact, the largest one. And he was working at one time on the Andromeda Nebula. This is the Andromeda Nebula right here. And he noticed inside the Andromeda Nebula that the nebula had a particular Henrietta Leavitt's kind of variable star inside of it. So he applied Henrietta Leavitt's ruler to the Andromeda Nebula. And you know what he found? He found this star was so far away, it wasn't in our own galaxy. Indeed, this star embedded in this nebula was outside our own galaxy. Indeed, this is no nebula, this is a galaxy. So Henrietta Leavitt's ruler enabled the Andromeda Nebula to become the Andromeda Galaxy, and in so doing, the world above our head became vast and ancient beyond description. And that was how important Henrietta Leavitt's work was. But Hubble took it one step further, along with other people, is he provided another measure. He used an idea that was around for a while. He was able to measure not only the distance of these stars using Leavitt's ruler, but how they're moving, the speed at which they're moving. And he discovered, now what we know is true, the expansion of the universe, that the universe is itself expanding. Well, if you take Leavitt's ruler and Hubble's expansion of the universe, with a few other discoveries that happened since, this is what really led us to think about the Big Bang. That the connections that we all share among one another, that we share with other entities, like the planet and the cosmos, really begins when the universe existed at a single point in space, and that has been expanding ever since. That's where the connections begin at this point in space, 13.6, 13.7 billion years ago. Now, since the Big Bang, in the early days of the Big Bang is an expansion of the universe. It's an expansion and the cooling of the universe. So what happens in the years after the Big Bang, say in the first several million years, is you have the expansion and cooling of the universe. And with that expansion and cooling, you see the successive formation of everything we find as familiar. First come the forces, which attract and repel the constituents of atoms as well as the atoms themselves. Then come the matter, which composes the subatomic particles. Then the subatomic particles, which themselves come together to form atomic nuclei, and atoms, and so forth. So really it's the emergence of the physical properties which make us happen successively after the Big Bang. Now the nuclei, the atomic elements of the Big Bang, really after the Big Bang, were really the smallest ones, the smallest constituents, the hydrogens and the lithiums. That when you think about, we learned about the periodic table in high school, and in junior high school. If you think about the periodic table that would have existed a million years after the Big Bang, that periodic table would have only had 3 elements on it, the lightest ones, because you really only had the smallest nuclei. It turns out that ever larger nuclei and larger atoms, if you will, are generated through fusion reactions that exist in stars. That is, as you learn to look in the sky, you see stars at different phases of their life history. Stars, the light of the stars and their heat, is generated by fusion reactions, where atoms, by gravity, are forced together. And as those atoms are forced together, they'll make a new, heavier atom. But in so doing, they'll release considerable amounts of energy. But as the stars consume one fuel, they start to make other kinds of atoms, and they start to recycle. They start to generate ever larger atoms. So the fusion reactions that generate the light of the stars come about through the gravitational pushing of atoms, like this deuterium and this tritium. The red is the proton, the white is the neutrons. They're forced together as a fusion reaction to make a larger atom with more protons. So there's 2 of these red things as opposed to 1, the guys on the left. And that reaction does 2 things. It releases energy, but it also generates ever larger atomic nuclei. So that's what happens inside of stars. So the central idea is that as the stars go through their evolution, if you will, they consume and generate ever larger atoms. Starting with the smallest constituents, those that began in the first million years after the Big Bang. So when you think about this is the periodic table, which some people love and some people run from in junior high school and in high school. But when you look at a periodic table, these atoms are organized by their atomic weight. On the upper left are the ones with the smallest atomic number, the so-called lightest ones, and the bottom right are the heaviest ones of the highest atomic number. Hydrogen and the ones with the lowest atomic number, the lightest weight, are the ones that are the nuclei that are generated in the first million years after the Big Bang. But the formation of the ever larger constituents of our bodies, the carbon, the nitrogen, the oxygen, and so forth, all the way up to iron, are formed inside the fusion reaction of ever different kinds of stars. Unfortunately, the physics of the situation is such that for generate atoms larger than iron, it requires ever more energy than the fusion reaction of a star could generate, and these are where the explosion of stars through supernovae generate the ever larger atoms and stars. The key point here is that inside our body lies the deepest material connection to the universe itself. That is, the atoms inside of us were born in the years after the Big Bang, or inside the fusion reactions or the supernovae of stars. The constituents that make us were part once of other worlds. And after we pass, those elements will be returned to the universe and then become part of other worlds after our life is over and after the life of our planet is over. So we are part of a grand cycle of the exchange of matter. Some of it derives directly, the lightest ones directly from the Big Bang, and others through this cycle of the birth, life, and death of stars. We, inside of us, have layer after layer of structure, just like we have layer after layer of history. And you can see that from our atoms to our cells themselves. Our atoms relate us to the cosmos. They relate us to the stars. They relate us to the Big Bang. Our molecules, and ever larger properties, such as cells and proteins, these relate us to the events in the solar system itself. And our organs and their structure relate us to our history with the planet. Now, I know this sounds strange, but let's go through it. How are we related to the solar system? What connections of the solar system lie inside our bodies today? So let's talk a little bit about that. And I'll give you 2 examples. Example number 1 begins with a brilliant, probably very eccentric Frenchman, Michel Sief. Michel Sief had a passion in life. And that passion in life was spelunking. He loved caves. And he also happened to be a first-class geologist. So he put those 2 things together and he investigated a cave. Well, one day in a cave, Sief had a brilliant idea. His idea was, what if I lived in this cave for, say, 2 months, 60 days, beyond time? No clock, no windows or anything like that. What happens to me? So Sief was a brilliant note-taker, because his scientific training, and he decided to set up an experiment where he would be his own lab animal. He would go into this cave for 60 days. He'd have friends on the surface with a telephone, and he'd call his friends at regular intervals. And he'd record when he went to bed, when he woke up, what he ate, when he excreted. He was a Frenchman, he recorded his libido, and all this stuff. And he had a telephone, and he'd call his friends, and he'd say, I'm going to bed now. He didn't have a clock, they did. So they'd record the real time of when he went to sleep. He'd call him up when he woke up. They'd record the real time of when he went to bed. Turns out, this experiment was supposed to go for 60 days. And Seif called his friends one day, and his friend said, Michel, how much lead time do you want to know before this experiment's over? He said, give me a couple days. His friend said, time to go. He had lost 20 days. And he lost 20 days because what to him, his perception of time completely went off. What to him were little tiny cat naps turned out to be full 12-hour sleeps. So he disconnected from the normal clock, but his body went to a totally different clock. What different clock did it do? It went to a 24-hour clock. He still had embedded in him a virtual 24-hour clock. Indeed, the more we look, the more we find each of us function on a clock. And you can see it in the physiology of our organs and the way our cells work. In fact, almost every process inside of us functions on a clock. The highest alcohol tolerance is right about now, so you expect. The lowest alcohol tolerance is in the morning, so it's probably the cheapest if you were looking to save money. Valves and kidneys are suppressed towards midnight. That's really good if you work in the Canadian Arctic in the summers. And the list goes on. Skid repair, pretty much all of our physiological properties function on the clock. Now, Seif did his experiment in the early 60s, and conveniently, by the late 60s, another kind of research was going on that not only linked to his clock, but linked to the solar system itself. And that research, of all surprising things, happened on flies. Why flies? Turns out the flies, if you want to understand genetics, are one of the best systems. And there's a lot of reasons for that. Flies breed fast, they develop really fast. You can get lots of flies, they're really cheap to work with. And you can breed them left and right and center, and you can look at their genetics. So for over 100 years, scientists who study genetics love flies, like this thing, Drosophila melanogaster. And the whole idea is, you could look for mutant flies, like this one. Right? Look for mutant flies, like this one with the extra set of wings. And you could breed it true, and if you're really good at what you do, you could find the gene that's driving this mutant. And once you understand the gene that's driving that mutant, you begin to understand what normal genes do. Okay, so it's really a fabulous system to understand genetics. In the late 60s, one of the lead laboratories for understanding fly genetics was at the California Institute of Technology in the laboratory of Seymour Benzer. Benzer was looking at fly mutants of all type, breeding them true and studying the DNA. He had a graduate student who found an odd fly mutant. This fly mutant had a weird rest-activity cycle. Yeah, flies have a rest-activity cycle. Part of the day they're active, part of the day they're not active. And he found flies where that rest-activity cycle was completely messed up. So he bred it true, like any good fly geneticist. And he found a gene that's responsible for that altered clock inside the fly's body. Well, that was huge, because finding that first gene enabled scientists to begin to pick apart the molecular apparatus that's underlying the clock inside the fruit fly. They found the proteins that the gene was specifying. They saw how that protein worked and what other genes it worked with. And they were able to understand the cellular clocks inside the body of a fly. So when you look inside a clock, like a Swiss watch, it's a complicated looking thing. But fundamentally, it has a kind of mechanical pendulum in it, where the timepiece goes, there's a back and forth cycle that dictates time. Turns out, the molecular clocks of Drosophila, they have a pendulum. They have a molecular pendulum, not a mechanical one, where the rise and fall of the activity of DNA rises and falls during a 24-hour cycle of the day. And it happens because of what scientists call a negative feedback loop. The DNA makes a protein, the protein downregulates that same protein in the cell of the fly. So it's kind of like a molecular pendulum. We can talk about it in question and answer if you want. But if you look at it, it's a lot of genes, a lot of proteins, about as undecipherable as a Swiss watch inside, to those who don't know the workings of the proteins. But the take-home message is the same. It's a molecular pendulum. But the take-home message for us is utterly profound, because every cell of our bodies, like that of Drosophila, and 2 trillion cells of our own cells in the bodies. Each one of them contains a DNA-based clock inside of us. So sitting here right now, you have, and I have, 2 trillion clocks ticking away inside our bodies based on the workings of DNA and proteins. These are incredibly profound. I pulled this slide from the web the other day, just to show you this one way. Which are you, an owl or a lark? Owls, we all know night owls, people who stay up late and go to bed, and who stay up late and wake up late in the day. Right? We call those college students, right? They stay up in the morning, they wake up at midnight. Larks are folks who like to go to bed early and wake up early. That's like me and my wife now. I'm a lark, so we kind of catch each other after dinner for a few minutes. Turns out that these aspects of this are actually encoded in the DNA. There's a syndrome called advanced sleep phase syndrome, where people who have this syndrome cannot stay awake past about 7 or 7:30 at night. And try as they might, they can't change their internal sleeping mechanism. And the reason is because it's encoded in their DNA. And the reason it's encoded in the DNA is it's a mutation of a particular activity of a particular protein and gene in their bodies. And that gene has been isolated in the last few years. Turns out that that gene, which causes advanced sleep phase disorders in humans, relates to a gene that causes altered sleep clocks in hamsters, and relates to the same kind of genetic anomaly that appears in the same gene of flies that are shifted in their rest activity cycles. So in all animal life lies clocks inside all their cells that's based on similar kinds of genes working in similar ways. And it's not just tuned randomly. It's tuned to a 24-hour, virtual 24-hour cycle that is inside our bodies, inside of flies, and inside of hamsters, and indeed all animals, lies a tie to the spinning of the Earth and our relationship to the moon in terms of the light-dark cycles which dictate our fundamental biology, as well as the 24-hour cycles that dictate the working of our genes and organs. So profound is the importance of this relationship to the solar system itself, and its origin and its working. Much of our health depends on it. The more we look, the more we find altered clocks lead to many different kinds of diseases, including susceptibility to diverse cancers. That's one connection to the solar system. There's one that's related to human evolution itself. If you want to think about our tie to the solar system and how it's influenced human history and human evolution, look no farther than work that was begun by the Pentagon in northern Greenland in 1959, 1960. Somewhere in the halls of the Pentagon, somebody had an idea to put a missile silo up here, and indeed an entire base under the ice to defend against nuclear attack, presumably from the Russians. And this project, called Project Iceworm, had in its base a whole city under the ice of northern Greenland called Camp Century. And what they did was the Department of Defense developed this giant cavern under the ice in northern Greenland. This is a schematic, it's seen in side view, of this entire city beneath the ice. Each of these lines here reflects a level, okay, a floor, if you will. So there are 7 or 8 floors. There was a mess hall. There was a nuclear reactor. There was a theater and a chapel, a library, an enlisted men's club. There was an officers' club underneath, all under the ice. The Pentagon planners got this thing going, digging a giant hole in the ice. And they discovered something that geologists and natural historians had known for a century or more. That, unfortunately, unknown to these planners, they would have saved a lot of money had they read their geology. Ice moves. They would have saved a ton of money for us U.S. taxpayers had they paid attention to the brilliant work of this great natural historian, Louis Agassiz. Louis Agassiz is one of the great figures in all of United States science history. He founded the Museum of Comparative Zoology at Harvard. He founded the Marine Biological Laboratories in Windsor, Massachusetts. Founded the National Academy of Sciences with Lincoln in the mid-1800s. But importantly, before that, he went on a summer vacation with his family in Switzerland, with his young kids and his family in Switzerland. And, they went to the Alps. And, the Alps is a really pretty place, see ice everywhere. But when Agassiz showed up, he ran into 2 friends. And these 2 friends were geologists who changed the way Agassiz saw the world. What they did is they showed him, they had a theory. They had a theory about ice. And they showed him the valleys of Switzerland, how the shape of the valleys in Switzerland looked like they should connect to the glaciers at the top of the mountains. They showed him erratic boulders that sat in these valleys, that they could not be transported there by any natural means. They had to have been moved by ice. So with those two friends, Agassiz was now able to see how these valleys were once filled with ice. And in that learning to see valleys, Agassiz and his friends saw the ice ages. They saw how what happens is, in these valleys, the ice was once filled the valleys and then later retreated. They saw the extension and retreat of the ice, which we now call, due to Agassiz's work, the ice ages. Agassiz extended this. This was in Switzerland. He began to see stuff like this everywhere, from England to closer to the Mediterranean, to the United States, that he really proposed, based on this, a very general theory on the ice ages, because he learned to see these valleys in an entire new way. Not everybody believed this theory, and indeed it was lacking a mechanism. And the mechanism was to come from some brilliant people looking at planets and the Earth in a new way. And part of the story begins with this guy, James Kroll, who was a brilliant Scottish mathematician. He owned a tea shop, so his point of a tea shop. I don't believe he said a lot of jokes to his customers a lot in that tea shop, but he was a brilliant mathematician. And he saw and proposed an idea that perhaps Agassiz's ice ages were based on changes of the orbit of the Earth. Just an idea that a Frenchman had proposed about 30 years before, but he really ran with it in a quantitative way. That work led to a fundamental breakthrough by this gentleman, Milutin Milankovitch, arguably one of the most brilliant and arrogant scientists of all time. He had a stated passion that he wanted to have a quantitative theory for weather. He used to chide his colleagues for having thermometers to measure weather. He believed that if he did his job right as a mathematician, he'd be able to predict the weather, that they wouldn't need a thermometer. Anyway, so Milankovitch ran with Kroll's idea, and he proposed a notion of orbital changes. That is, every 100,000 years, the Earth's orbit changes from being more circular to being more ellipse. And then on smaller cycles, say every 40,000 years and every 20,000 years, the wobble and tilt of the Earth changes. So what he saw and proposed was that the Earth's orbit changes in regular ways on the orders of 100,000 years, 40,000 years, and 20,000 years. That these are cycles. And that these cycles would interact with one another such that every now and then they'd superimpose in such a way that they'd cause major climatic changes on the planet, which would lead to the ice ages. So he proposed this brilliant notion that the Earth's orbit changes in regular ways, and that those regular variations affect the waxing and waning of the glaciers. At the root of these changes were what he saw as the interaction of the Earth and its orbit to Saturn and the other planets of the solar system. Because each of these planets is exerting a pull on the Earth, right, a gravitational pull on the Earth. And they'll do so in regular ways. So one outcome of the interaction of the Earth with these different planets is regular variations of the Earth's orbit, which would change the waxing and waning of the glaciers over time. It really wasn't until, Milankovitch did his work in the 40s, it really wasn't until we were able to date the ice, and actually some of the first ice to be dated geochemically was in Camp Century in the deep hole that the Pentagon so graciously dug for the scientists. But in doing that, in dating the ice, and in looking at the predictions of Milankovich's theory, it was found there was a broad correspondence between the predictions that you would of the waxing and waning of the ice based on this theory of changes of the Earth's orbit, and the actual dating, the timing of the waxing and waning of the ice. Well, if you look at the ice ages, which are driven in part by these orbital variations described by Milankovitch, they have been important factors in human history and in human evolution. I mean, if you look at human evolution, one of the great shifts that led to the success of the human species is a transition from woodlands in Africa that existed about over 3 million years ago, to open savanna plains that existed ever since. That transition from closed woodland to open savanna plain is one of the transitions that is thought to allow the flourishing of the bipedalism